Okay, I'm recording now. How about other questions on chapter 4? Or?
So it's 1, 3, and 4.
Yep. With 4 having so many different topics, I mean, it's got tons of different things in it. And I know that makes it a little more challenging sometimes. 1's really shouldn't be too bad. 1 shouldn't be too bad. We didn't do a whole lot in 1. 3 was the cell, right?
Yeah.
Cell organelles, the cell cycle. Trying to remember what else was in that chapter. I would think 4 was probably one of the more challenging chapters.
Okay.
I don't know if you agree,
Right.
but yeah. Okay, I'll put whatever we talk about, in the review if you can't stay.
Yeah, I gotta go. Hey, thanks, guys.
Okay, John, see ya.
Right.
Bye, John.
Right.
So Jolene, Sadie, other topics and
So basically, like, my question is, like, how, like, will it be distributed, like 1, 3, and 4? Like, are you, like, not going to focus on 1 a lot because it's like an easier chapter? Like, how will it be?
yeah, good question. I think it's probably safe to say there won't be quite the same emphasis on 1 as there will be for 3 and 4, because as you say, we really, it was an introductory chapter. We didn't get into any of the anatomical terminology, the body cavities, membranes, the regional terminology. That was, of course, covered in lab. So there's a big chunk toward the back end of it that you don't have to worry about for the lecture exam. And then the first few pages was just more of a general introduction into the field. So in terms of what we talked about, homeostasis was a big part of that. There was that hierarchical figure where we started with molecules and worked our way up to organism. There was a question on the quiz about that, so that would be a good figure to look at. But I think if I had to pick a topic that's really important, it's the homeostasis and the role of receptors, control center, and effectors. Yeah, that's a critical part of that chapter. You will see stuff on that. Oh, and then I guess we did talk briefly, or I had you look over that table where it listed the 11 organ systems. And there was a question, I think, on the quiz about, name 2, give me structures and general functions. So that would be good, of course, to go back and look at. But yeah, that would pretty much be 1, I think. And then 2, of course, we have the chemistry test. So there's nothing on 2 on next week's, or this week, I guess now, Wednesday's test. So I would hit 3 and 4 hardest. I would not forget about 1, but yeah. 80 points. I think I told you guys kind of the format. Multiple choice, probably about 30 to 35 multiple choice, each worth 2 points. Somewhere in the 10 to 15 matching number, 2 points each for those. And then your choice of, I think, 2 out of 3 essays, each worth 5 each. So it's like 20, 15. That would be 35 times 2 is 70 plus 10 is 80. Yeah.
So for the essay questions, like, what could be like a subject maybe that you said there's.
Will you tell me, what do you think would be a good essay question?
I'm not sure. Maybe what we just went over. I know you said that there's going to be choices.
Yeah.
Okay.
Because the reason that I asked you that is not to put you on the spot, but to have you begin to think about, hey, what might he ask? What would be a good essay, And if you can come up with that question yourself, then you've answered it. So
okay.
So here's a couple examples. I could ask something like, name 8 of the 11 organ systems, or name all the organ systems.
Okay.
And rather than an essay, maybe you would put that in a table format. So
Okay,
that's not technically, I guess, an essay, but that could be that type of a question you could be asked.
okay,
Um,
that clears it up, because I was thinking like we're gonna have to write like a whole essay, like like an essay
well,
to
for
a
that,
question explaining, but
yeah, for that particular type of question, it would be a heck of a lot easier for me to grade and a lot easier for you to organize it if you did a table.
okay,
But an essay that might involve some writing could be like, describe the 4 stages of mitosis
okay,
using sketches, labeled sketches, what is going on during each stage. So what are the 4 stages? You should know this.
Prophase, metaphase, anaphase, and telophase.
Right, in that order, right.
right, okay,
Yep. We just did a little bit of that last week in lab.
right,
So you've heard it and seen it, actually. I think you all saw some chromosomes last week. So that'd be a good type of essay question. I don't generally ask you to sketch a lot of things, but I think that could be pretty, I won't say easy. I'm sure I can do it. Yeah. What else? I could ask you, or I could give you a strand of DNA, and maybe ask you to indicate what would the messenger RNA base sequences be if this was the template strand of the DNA you were using. So you'd have to say, of course, opposite A is a U in the case of RNA, and then opposite G is C, and so on and so forth. Or I could give you the messenger RNA strand and ask you, what would the linear sequence of anticodons be to this messenger RNA strand? Now you have to remember, a codon is a 3-base sequence, so I might give you, let's say, 5 codons, which would be 15 bases, and ask you, what the anticodon sequences would be. I'm not gonna ask you to tell me what would the amino acid coded for UCC be. If I did, I'd give you the table. So don't worry about memorizing this table. But now you know how you use it, right? We just did some examples.
right,
And note that these 3 guys here are called stop codons. They do not code for any amino acid. So what do you think these do?
Are they like the end of the DNA?
No, this isn't DNA. This is messenger RNA.
they stop it?
They
Is
stop.
that
They stop translation. Exactly.
Right,
In other
Was that
words,
like breaking points?
well, they're like the end of the road for the train. We've gotten to our destination. We don't go any further. We're done adding amino acids. We've made our protein. Basically, that's what that means. If the see the ribosome, what it does is it slides across this messenger RNA, and when it reaches one of those stop codons, then no more transfer RNAs can fit in, and we're done making the protein. And so I think what I talk about in the chapter in my lecture PowerPoint is what happens if you were to introduce a change in the DNA sequence. Right? This is discussed here in the PowerPoints. If you change a base in the DNA, in this case, you change it from T to A, then what that can do is alter the codon in the messenger RNA such that a premature stop codon could be introduced by the mutation. What effect would that have on the protein being made? if you had a premature stop codon introduced? Would that be good or bad?
Bad. You wouldn't have all the DNA.
You wouldn't have the protein properly constructed. Remember, these code for amino acids. These are the building blocks of proteins.
So like when you have like an extra chromosome, then is that like the stopping enzymes like they like are late then?
No. When you say extra chromosome, that's totally
Is that
different.
totally different? Okay.
That's that. I mean, you're thinking at the right genetic level, but we're talking about here a very small part of a chromosome, like part of a gene.
Okay.
But what I want you to kind of think a little bit about is if you lay out in the sun a lot and you get skin cancer, because there is a connection between the two, right?
Mm-hmm.
That long-term exposure to the ultraviolet rays of the sun can and will over time increase the risk of mutations in skin cells. And that can lead to cancer. Now, we haven't talked about cancer here. We're talking about what does a single point mutation, it's called, where we're changing one of these bases. What does that do to the protein being made? But you've learned back in one of the chapters, I don't know if it was this one or the preceding one, that the process of cell division, I think it was chapter 3, is well orchestrated. There's green lights and red lights that the cell gets within the cell cycle. Do you remember hearing about that? Back in chapter 3. Go back and review it.
Yeah, and then so if it just like gets immediate green lights, that's like cancer, because then you just like you had to think like a dish of like skin cells.
Yeah.
So I just like kept reproducing. So it's just like a bunch of green lights.
Exactly. A bunch of green lights means the cells are dividing. There's no control. They just keep on dividing, and you got a tumor. Exactly. So mutations are not super common because what our cells can do, it's amazing, but they have proofreading enzymes that will take a piece of DNA that's just been replicated, and these enzymes will scour the newly synthesized replicated DNA, and if they find a mismatch, they'll take out the wrong base and put the right one in. They'll fix the error. These errors happen very, very rarely. And when they do, they're typically caught. But as an individual gets older, the system, we'll just say, isn't as efficient. And so sometimes some of these things can escape detection and can end up turning what would ordinarily be an amber and then a red light, right, to a constant green. Yeah. So that occurs as a result of age. It occurs as a result of exposure to mutagens, they're called, things in our environment that can cause mutations, like sunshine, like certain chemicals. And benzene is one that I remember when I was in college, we used benzene in organic chemistry lab all the time. It had a very sweet banana odor to it. I'll never forget. And we had to always use hoods, which are there are things in the lab where you pull down a big, glass front, and you work underneath it, and all the air gets sucked out, so you're not exposed to the chemical. So all of our experiments with using benzene were done under hoods in the chem lab. Now they don't even use benzene, I don't think, in organic. It was a known carcinogen. It's nasty to be exposed to because there was definitely an increased risk for problems down the road if you work with benzene a lot, even smelling it, I guess. So, we face a number of mutagens out there that can cause problems. Even getting exposed to certain viruses can cause cancer. Did you know that? Yeah, it has nothing to do with the food you eat or the air you breathe. You just catch a virus.
Wow.
Yeah. I mean, you've heard of the human papilloma virus that they're inoculating kids with before they become sexually active to reduce any chance of cervical cancer.
Yep.
And of course, that's a big debate about, would you give your 12-year-old, would you make her have a shot Well, the answer for me is absolutely yes. I don't mind getting cervical cancer. If I can stop that or prevent that,
Right,
why the hell wouldn't you? I mean,
right.
I'm stepping out of bounds by probably getting so worked up about that or whatever, but it bothers me when you can utilize the advancements of science to help, and some people just will not do it for lots of different reasons, and I got to be respectful of religious, I guess, viewpoints, but anyway, yeah, it's pretty interesting stuff. Other topics?
That was most of my questions that I wrote down.
Great.
And mine were the same as hers, coincidentally. Yeah, I was like, wow, okay, somebody else is asking the same thing. Okay,
Yeah, that's not
yeah,
unusual that you'd both be
Yeah.
asking about
Yeah.
that. Good. Have you tried to play some of these videos, and have they been working okay?
yeah. I've watched under wherever it is in Blackboard a few of your videos. Like I've watched the first two twice just to kind of like digest it, hear it, and then as I'm going through my like study guidelines, I'm listening to it, reading, trying to.
Oh, I guess you were thinking of my Zoom recordings, lecture
Oh,
recordings,
yeah.
probably, right? I'm sorry, I was referring to the embedded videos in the PowerPoint. Like sometimes in the past, students have had problems playing those. I always try to make sure that they play okay for me when I review the PowerPoint before lecture.
Okay.
But if you ever encounter, like, say, this slide doesn't play properly, let me know.
Yeah, I haven't had any problems.
Okay, good. I was preparing for my microbiology Zoom lecture just before I came on with you guys, and one of the links I just clicked on the hyperlink to make sure it worked, and it said video has been removed from YouTube channel or something. So I had to, try to find a substitute. So once in a while, for whatever reason, these links sometimes go bad. But a lot of this stuff you can find. I mean, YouTube's a great place to look for videos. The Khan Academy, if you've ever heard of Khan Academy, that's a lot of good videos there on science related stuff. Really good stuff.
Yeah, for the quiz, I watched a couple YouTube videos while I was just on the treadmill.
Oh yeah, good.
This-
Are you doing it at home or at the Y?
That's why.
Uh-huh. Yeah.
I'll just set it up because I'm on there for like 40 minutes because I just do like the walking incline. So I'm on there for like 40 minutes so I can get like 2 videos in about.
Good for you. Good for you.
It's actually a really good idea. I go to the Y to use the treadmills too, so
You did?
maybe that's what I'm saying. Yeah.
I've seen you there and didn't even recognize you.
Yeah.
Do you have to wear masks? You do, don't you?
Yeah.
Yeah, sometimes it's hard to recognize people, even,
Yeah.
So do you have earbuds, and can you hear what's going on too then?
Yeah, I have my AirPods in.
Yeah. It's wireless or not?
Yeah.
Great. That's nice.
Yeah.
You almost have to make the most efficient use of your time, and that's really great. I'm sure as you listen to my PowerPoints, you just like you're running faster. It's easier, right?
Yep.
All right. Well, I will let you guys go then, and we'll see you Wednesday.
Okay.
Sounds good.
Thank you for your
Okay.
time.
You're welcome. Have a great weekend.
Thank
Sure.
you.
Bye.
Bye.